There we go. We're recording. Good timing. So that's how that's going to work. Like I said, if we don't finish the exam, if we don't get to the exam review today, I'll pick a time tomorrow. We can do it then if you want to. So let's get started. I'm glad you guys are all online. Let's jump into chapter 3. Let me share my screen. Okay. You guys should be able to see my screen by now. And at this point, you should be able to, where are we at? See my PowerPoint. So if someone cannot see my PowerPoint, let me know. I would say if you can't hear me, let me know. But obviously, if you can't hear me, you wouldn't hear me say that. So here we go. We were talking about proteins when we finished last week. I backed it up a little bit to remind you of what we talked about. The first thing we talked about was the monomer proteins, which are amino acids. And you need to know that. Remember, you need to know that for all these things. You need to know the parts that make them up. Carbohydrates, the monomers are monosaccharides, and the polymers are polysaccharides. Proteins, as you know now, the monomer is amino acids, and they make up something called, well, I'll come back to that. We haven't told you that yet, but it's polypeptide. We'll talk about it here in a second. And remember, lipids aren't necessarily monomers made up of polymers, but we did say, for example, triglycerides are 3 fatty acids attached to a glycerol molecule. And we know for steroids that those 4 rings is how you would identify those. So anyway, about the proteins, we talked about amino acids. I said that you need to know that there's 20 of them, and everything that's alive, all the protein that's built in every living thing is built from these 20 different amino acids. I said you did need to know that. You do need to know the structure. You need to know that it's each one is a carbon attached to an amino group, attached to a hydrogen, attached to a carboxyl group, attached to a side chain. You do not need to recognize this, so I'm not going to show you a picture of this without the words and say which one's the carboxyl group and which one's the amino group. If you ever did get that question, though, the easy way to tell is the carboxyl group has a C, so does that. The amino group has an N in the word, and so does the amino group. So anyway, know the parts to the amino acid. All amino acids have the same exact shape or structure, if you will. The only thing different for each amino acid is the side chain. So the side chain is what defines the amino acid, so to speak. Because, and again, this is a review from everything we said on Friday. Because this part right here, right, this is the part that is exactly the same on all 20 amino acids. That part is exactly the same. It's this side chain that is completely different for each amino acid, and that's what defines it. So keep that in mind. Let's move forward. That's where we left off. Then we said that these amino acids, each one of these 20 different versions of amino acids come together, right? And when they come together, that is called a peptide bond. You do need to know that. I don't know why. I mean, I kind of know why, but I don't, in my opinion, it's not that important because a peptide bond is a specific type of covalent bond. So it's still a covalent bond. It's just specifically a covalent bond between 2 amino acids. And I guess one of the more helpful reasons that you should know that is because then that will help you remember the name of the polymer, of a protein, which is the polypeptide. So remember that for the exam. The monomer is the amino acid, and the polymer is the polypeptide. So then the question is, what is the difference then between a polypeptide and a protein? And this is where things get a little bit interesting and a little bit more complicated. I don't remember if I did this example on Friday. Did I fold any paper on Friday that you guys remember at the end of the lecture? Because this is where we were, was at the end. I guess not. No. Okay, so I'm going to do this very quickly. Well, what do I have here? Can someone tell me what that is? I mean, in general, not specifically what it is.
Paper.
Yes, it's a piece of paper, right? That is a piece of paper. Bear with me for a second. Should have done this beforehand. See, I guess here's an advantage of being online. You guys can't see what I'm doing. I'm sure it's so exciting, all the suspense. Anyway, can someone tell me what this is?
Paper airplane.
Yeah, it's a paper airplane. But technically, it's also still a piece of paper, right? Would you agree with that?
Yes.
Okay, and that is the relationship between a polypeptide and a protein. So a polypeptide is just a chain of amino acids, right? It's a bunch of amino acids that have been added one onto each other. And it's just that. It's just a polypeptide until it folds up on itself and gets it into its final shape. At that point, it's considered a protein. Once it's folded and it's in its final shape, it's considered a protein. Yeah, technically it's still a polypeptide, but it's not a protein until it gets folded and meets that shape. Just like that piece of paper was a piece of paper until I folded it into this shape. Now it's a paper airplane. Yes, technically it's still a big piece of paper, but now that I folded it into its final shape, it is considered a paper airplane. So yeah, that's an important concept, an important relationship that you need to know between polypeptide and a protein. I also use this picture, and this one's a little bit more realistic because when you think about a chain of polypeptides, it's going to be that, right? A long chain. Now this is just a big flat piece of paper, but yarn is similar to that, right? It's a long thing. It's a long string. So this thing right here that you see, I guess that's a blanket maybe somebody knitted. Technically, that is a piece of yarn, right? It's a piece of yarn. It's not considered a blanket until it's twisted and folded and into its final shape, right? It's still a piece of yarn, but now that it's into that twisted and final shape, we now consider it a blanket. So that is the important relationship between a polypeptide and a protein. So here we go. This is a picture of what we were talking about. We have this amino acid coming together with this amino acid. Not that you need to know this, but it's always going to be the carboxyl group that bonds with the amino group, which makes sense because remember, something you do need to know for the exam is when you bring two things together, that's always a dehydration reaction, right? Because the dehydration reaction forms a water molecule. And in this case, you can see we have an OH here and an H there. So you bring those things together and you get a water molecule. But again, most important thing that you need to know for this is just a reminder that when we bring two things together, and this for this exam, actually for this entire semester, when we bring two molecules together, we call that a dehydration reaction because we've basically taken water out of the reactants and produced water, in the products. And yes, technically this covalent bond, we call that a peptide bond because it's not just any old covalent bond. It's a covalent bond between two amino acids. So any questions about this picture? Okay. Let me say this. I know you're all online, and I'm not saying this. I'm not talking bad about you. I'm talking bad about me. I've mentioned this before. I've been on a lot of online meetings, and I'm a busybody. So even though I think I'm paying attention and I say I'm paying attention, I also have my computer in front of me. So I'm doing other things. So if that's you, try your best not to. If that is you, if that's you, try your best not to. But also be honest with yourself. If you're that type of person, even though you may be here and present, you might consider going back and watching the video. If you can be honest with yourself and say, I kind of dazed out for 10 minutes there. So anyway, let me move forward. So there's thousands of different proteins in the human body. That's a good independent work question. Look that up. Exactly how many proteins are there? I shouldn't say exactly because we don't know exactly, but we have a pretty good ballpark figure. So the question is then, if humans have all these thousands of different proteins, but we only have 20 different kinds of amino acids, how is that possible? And your book uses a great example. So think about just a book, a really thick book, not even a really thick book. Think about like a 100-page book. Here you have the story, 100 pages long, and you have all those different words made from only 26 letters in the alphabet because it's all in the different ways you put the letters together, right? If you put those letters together in different ways, you get different words. And that's kind of the same thing here with proteins. You can think of each amino acid as a letter. Of course, these words are going to be a lot longer than the words in the English alphabet or in the English language, but that's how you can think about it. So yes, there's only 20 kinds of amino acids, but there's all these different combinations of ways you can put them together. And because of that, we have thousands of different proteins. But again, for the exam, the only important thing you need to know is what I've already told you, which is that there are 20 different kinds of amino acids. So your book, I use this as an example. Again, so there's 1 million, as of when I made this PowerPoint, there were 1,025,109 words in the English language. All of that from 26 letters of the alphabet, right? And remember, with the letters of the alphabet, there's rules. Like, well, you have to have this before this, and there's always a U after a Q, right? So you have all those rules you have to deal with. With proteins, there are no rules as far as that, right? Those 20 amino acids can come together in any combination. So any questions about that? All right. If you download this PowerPoint, this is a video you can watch. This is a polypeptide. It's not necessarily a protein yet because that's just the string of amino acids. And what the video shows is that thing folding up on itself and getting into its final form. And again, at which point that is when it's called a protein. So what determines that final form, right? Because in a way, when I was using that yarn as an example, that was in one way it was a bad example because I could put that yarn in and do anything with it, right? I can make a scarf out of it. I can make a blanket. I can make a sweater. I can just ball it up, right? But with amino acids, it's different. With proteins, it's different because each amino acid, the sequence of the amino acids, that determines how that thing is going to fold up on each other. And I mentioned that before when we were talking about when I showed you those two amino acids. And there was two different side chains. And I said, remember, this one's hydrophobic and this was hydrophilic, I think was the example. So those things repel each other. So if they're on a chain, I wish I had a chain with me, but if they were on a chain and you try to fold that chain up on itself, and this is them, they're not going to go, right? They're going to repel each other. So that chain is not going to go that way. It's going to go away. So it's things like that. You've got things that are attracting each other, things that are repelling each other. And that's what gives it its three-dimensional shape. This is an example that's not from your book. Here's a string of amino acids. This is technically a protein because it has folded up on its final shape. But it's a very simple one because there's not a lot of interaction going on. As far as this picture is concerned, the only interaction you see is between these two amino acids. Then they're both the same type of amino acid. So here's a cysteine, here's a cysteine. Each one of them, for their side chain, they have a sulfur at the end. And those sulfurs are attracted to each other. So because of that, this amino acid has a curve. You can see where it curves, right? And the reason it's curving is because these two things have covalently bonded to each other. That's just an example. You do not need to memorize any of this. The point is all those different side chains for those 20 different amino acids, because they have different shapes and sizes and chemistry and charges, they repel and they attract and they do all kinds of different things. And that's what gives a protein its shape. Which is important because later in the semester when we talk about genes, then this will be very important because, or when we talk about making a mutation, if there's a mutation, that means you change the code. And if you change the code, let's say, for example, I shouldn't get into this too much because this is ahead, but if you were to change this amino acid to a different type of amino acid, then you would also get rid of this bond, and therefore this string, instead of curving, might just go straight out. But just an example. Again, another example. This is everything I've said, but in a picture. Here on the left, we have ourselves a polypeptide. It looks like it's folded, but it's not. It's the artist just put it that way so we could see it all in one picture. But it's just the amino acid sequence. It's just a polypeptide. It's not folded into its final shape. But because of all the different interactions in between those amino acids, which we can't see, they don't show them, it does fold. And you're not expected to understand what this means, but there's two different ways of looking at it. This is called the ribbon view. So this is what the molecule looks like when you portray it that way. This is another way of portraying it. But the point is, that's a very distinctive shape, right? Which either one you look at, you can tell it's a very distinctive shape. And again, the point is, the reason it has that shape is because of all these different interactions between the 20 different types of amino acids. So any questions about that? All right. As I've kind of alluded to earlier, if you have a slight change in the sequence of amino acids, right, like I pointed out earlier, if you change that cysteine, then that thing's no longer going to be attached to that. That will affect the ability for the protein to function. So here's something you need to know, and you can write this down if you're taking notes. A protein's function is determined by its shape. So I know we've been talking about structure and function this whole chapter, and even a little bit in the last chapter. But of all the molecules we talk about, proteins are the most important when it comes to structure and function. It's all about their shape. Proteins work because of their shape. If you change their shape, you change the way they work. And knowing that, and knowing what I just said, which is that it's this sequence of amino acids that gives them their shape. If you change the amino acid, you change the shape, therefore you change the function. And your book gives a good example of hemoglobin. So if you have one substitution, like one genetic mutation that we'll talk about later, you change this one letter, then you change one amino acid out of a whole long chain of amino acids, right? You just change the one. And when that happens, here it is right there. You can see where it is. You change the one amino acid, and because of that, the whole polypeptide does not form properly, right? It doesn't fold on itself the right way. And because of that, the whole protein is a little bit misshapen. And because of that, that one protein, the whole blood cell is misshapen. And that's where you get sickle cell anemia, right? That's where this disease comes from, is because one letter was changed and you changed one amino acid. you change the shape of the protein. And it's a very important protein. That's a protein that carries oxygen, right? Without that protein, people die. And that's why sickle cell anemia can be deadly. And we'll talk more about that, I think chapter 10 when we talk about mutations. So we'll talk about that later. For now, this is just an example. You don't need to know anything about it on exam 1. But are there any questions about that? All right. This is everything I've said, but now it's in writing. You can see the words. One difference, unite, right? If you change out one amino acid, if there's one difference, that's enough to cause the protein to fold into a different shape. Again, if it has a different shape, that means it has a different function. Sometimes that just completely destroys the function altogether. Either way, that'll cause, could cause diseases. And misshaped proteins are actually associated with a whole line of different diseases, one of which are called prions. And there's questions about this on the study guide, but I don't think I'm going to ask. Nah, I almost guarantee I won't ask anything about them on the exam. But you have probably heard of prions. They probably just weren't called prions. You probably just heard one specific type. And here's the example that most people I think have heard of is bovine spongiform encephalopathy, which is basically, it's mad cow disease. So what we mean by spongiform, it means like think of a sponge, right? You have a bunch of holes in the sponge. Encephalopathy, basically we're talking about your brain. So what this means is basically your brain turns into a sponge. You get a bunch of holes in your brain. And the bovine just means cow. So anyway, mad cow disease, yeah, that's just a bunch of holes in your brain. And that's because you get these proteins that are misshaped. And like you can see here, this one, it's called a prion. It's a misshaped protein. When it touches another protein, then that protein misshapes, right? And then it touches another protein and that one misshapes. And that's how you get these holes in your brain. And again, the most famous example, I think, is mad cow disease, but it happens in a lot of other types of animals, including humans. Kuru is a good example of that. That's interesting if you want to look it up for independent work. Usually that was passed along by cannibalism. And there are groups of people in the South Pacific that have very recently still practiced cannibalism. So the disease is still around. Anyway, that's what a prion is. It puts holes in your brain. It's deadly, and it's from a misfolded protein. Any questions about that? All right. So again, let me reemphasize this point. A protein's shape is so important, right? But without that proper shape, it's not going to do its job. So that being said, it's also important to know that its shape is very sensitive to the environment. One thing I haven't told you yet is that generally speaking, when there's a bond, whether it be a hydrogen bond or an ionic bond or a covalent bond, it takes energy to break those bonds apart, right? It took energy to build it, but it also takes energy to break that bond. So what happens here, this is a good example of a protein. We can see all these different amino acids. This particular shape of the protein is a spiral. And what these little dots represent are hydrogen bonds in between this amino acid and that amino acid, this amino acid and that one, so on and so forth, right? Remember, hydrogen bonds are the weakest of the bonds. So I don't expect anybody to remember this, but as a reminder, oxygen is red, right? So we know that thing is partially negative. Hydrogen is white, so we know that thing is partially positive. There's actually a different symbol for partially negative and partially positive, but we're going to go with that anyway. So yeah, you can see these things are hydrogen bonded to each other because, this is slightly negative, slightly positive. But if you change the environment, you could break those bonds. Like I said, it takes energy to break bonds, right? So one way you could change or break the bond is something I've already told you about: temperature. Temperature, when I say something's getting hot, remember I said this, I think, last week. When something gets hot, what you're really saying at the molecular level is that the molecules are moving faster, right? There's more energy in the system. Whether you're talking about hot air, that means it has more energy. Hot water, it has more energy. Hot metal has more energy, right? Hot means more energy. So if you put energy into a system, or in this case, if you put energy into whatever this protein is in, then it's more possible for those hydrogen bonds to break because there's energy now to break those hydrogen bonds. Same way we talked about water, how at first it didn't change temperature because those water molecules are bonded to each other, hydrogen bonded to each other. And it isn't until you break those hydrogen bonds that the temperature really starts changing. So again, think about that with the protein. If you change the temperature, you're going to change the shape. Likewise, if you were to make it smaller, these things, that bond might get a little bit smaller itself. They might be, more attracted to each other because there's less energy trying to push them apart. Another thing to consider is pH. So if I say something is acidic, and you should know this for the exam, so hopefully someone can answer this. If I say something is acidic, like if I say this protein is in an acidic solution, what am I saying? What is in that solution? Obviously acid, but how do we define acid? What kind of ion is in that solution?
Hydrogen?
Yes, specifically H plus, right? Because the hydrogen is just the atom, but an H plus, that's the ion. So yeah, if something is acidic, now this thing's fully charged, right? That's a full positive charge. This one, even though I drew it as a positive, it's only partially positive, right? So if you put this protein in acid, it's going to be surrounded by a bunch of fully positive hydrogen ions. So this negative oxygen is going to be fully attracted to that fully positive hydrogen, right? It's not going to care about that slightly positive hydrogen. Therefore, the whole thing is going to unravel. And the opposite would be true if you put it in base, right? Because then you have the hydroxide ion, and that's fully negative, right? So this hydrogen here, it's not going to be attracted to this slightly negative oxygen anymore. It's going to be attracted to that fully negative hydroxide ion. All this to say, the only thing you need to know for the exam, again, the shape of a protein is very important, and its shape is very much dependent on its environment. And two great examples that I've just spent too much time talking about are temperature and pH, right? It can cause things to unravel. It can cause them to change shapes. So are there any questions about that?
I have a
Good.
question.
Oh, sorry. Question?
So you're saying that if there's a change in pH in like muscles, that can like cause big problems?
It could, yeah. It depends. Yeah, exactly. It depends specifically on the exact structure of the protein and exactly how much your pH changed. But yeah, it could be big issues. And now you can see why, right? Because,
Yeah.
in this example, the ions will disrupt the hydrogen bonds in that one example. And there's a good question. I'm glad you asked. There's a good question on your study guide that talks about a bacteria that lives in 100 degrees Celsius. And if you guys aren't familiar with that, that's boiling point, right? So it says you've got this protein from that bacteria that's used to 100 degrees Celsius. You have a protein from a human, and you put them both into really hot water or something, and which one maintains its function, I think was the question. Obviously, it's only going to be the bacteria, right? Because the bacteria, it lives in 100 degrees Celsius, so all of its proteins are used to being that hot. Meanwhile, a human would be boiled to death in that temperature and would die, not just from being burnt and boiled, but because the proteins would unravel. And the word for unravel is denature, not that you need to know that. So any other questions about proteins? Sweet. All right, let's move forward. We're going to go through nucleic acids very, very quickly because we're going to have multiple chapters where we talk about nucleic acids. So in my opinion, this is just an introduction. So I'll tell you all the important stuff that you need to know for the exam. Or if you've already done the study guide, you probably already know, because anytime you miss a question, or anytime you answer a question that's not on the exam, after you've answered it, it tells you, right? It says, hey, by the way, this is not on the exam. But anyway, nucleic acids, what do? They store information and they provide instructions for building proteins. You do not need to know that for this exam, because we're going to talk about that a lot later in the semester. You do need to know that there's two types of nucleic acids. There's DNA and there's RNA. We're going to talk about those both in depth a little bit later in the semester. But for now, that's all you need to know. It's also worth noting for our later discussions that there are different types of DNA. And there's many, many different types of RNA. But anyway, when you look at the big umbrella blanket of nucleic acids, there's two categories, DNA and RNA. So you need to know that. Technically, DNA is called deoxyribonucleic acid. We'll discuss that later in the semester. Technically, RNA is called ribonucleic acid, and we'll discuss that later in the semester. So any questions so far about nucleic acids? Okay. Gene. We're going to talk a lot about this later in the semester. There's a question about this on the study guide. So I'm going to go ahead and discuss it very quickly. But for this chapter or this exam, you don't need to know anything about a gene. But a gene is the unit of inheritance that is encoded in a specific stretch of DNA, and it programs the amino acid sequence of a polypeptide. An easier way of thinking about that is think about this long strand of DNA, right? And if you were to zoom into that long strand of DNA and just look at one particular strand, it's going to have codes, right? A, T, C, G, all these letters that you're going to learn about later. And that code says, all right, here, give me this amino acid. Okay, next, attach this amino acid to that one. Okay, next, attach this amino acid to that one. That's all the code does, right? It says, tells you which amino acids to put together. And at that point, once you have all the amino acids you need, that thing folds up and then you have yourself a protein. So a gene is basically, it's nothing but a set of instructions on how to build a protein. It's essentially that simple, at least in this class. One thing you do not need to know for this exam that you're going to learn a lot about later in the semester is this set of instructions in the gene, in the nucleic acid, in the DNA, that's written in a chemical code. That is the DNA. That has to be transcribed, right, from DNA to RNA, and then later that information has to be translated from the nucleic acid language, so to speak, to the protein language. So everything I said earlier when I said, bring this amino acid in to this amino acid to that amino acid, that would be translation. And again, we're going to learn about that later in the semester. So as far as the exam is concerned, this whole slide is nothing but an introduction, and it will not be on exam 1. Are there any questions?
Do we need to know like the definition of a gene for the exam?
No, nothing on this slide will be on exam 1.
All right.
Including the definition. So good question. And here's everything I said, but in a picture. Imagine again, this DNA strand just keeps going this way and keeps going that way. Here we've zoomed into a particular strand of the DNA, and it's a gene. And what happens is that information it's read and then written onto a strand of RNA. And this is very important when we talk about COVID vaccines, but we'll do that later. And then that information is then translated into amino acids, which then fold into proteins. So again, this is just an introduction. You're not going to need to know that for the exam. What you do need to know, as I said this at the beginning of the chapter, and keep saying this throughout the chapter, you need to know the monomers and the polymers of everything we've talked about, right? So again, the monomer for a polysaccharide is a monosaccharide, right? The monomer for a protein is an amino acid. The monomer for a nucleic acid is called a nucleotide. So make sure you know that. What is the monomer of a nucleic acid? It is a nucleotide. And just like you needed to know the different parts of the amino acid, you also need to know the 3 basic parts of a nucleotide. And technically, it's a 5-carbon sugar and a phosphate group and a nitrogen-containing base. But I put in bold what you should remember, right? So if you're taking notes, if you can just remember sugar, phosphate, base, you're good. So know those 3 parts. Sugar, you can see here, phosphate, and a base. And just like the amino acids, remember amino acids, we talked about 4 parts. Well, 5 if you include that central carbon, right? The amino group, the carboxyl group, the hydrogen, and then there was that side chain, right? And that side chain was the only part that was different. So it was the side chain that defined the amino acid. So with the nucleotides, it's similar. No matter what, the sugar and the phosphate's the same, but it's the base. The base is the only thing that varies. So it's the base that it's the base that you can identify with the nucleotide. So this particular nucleotide in this picture, that's not from your book, is called cytosine, right? Because it is defined by this base. Doesn't matter if it's a C, a T, an A, or a G. This part, the sugar and the phosphate's all the same, but this thing is a cytosine because it has that base. So any questions about this? Again, you need to know for the exam that nucleotides are the monomers of nucleic acids. The 3 parts of a nucleotide are sugar, a phosphate, and a base. And the base is the only part that varies. It is the base that defines the nucleotide. Here's some more pictures. This one's actually from your book. Again, here's the sugar, the blue part. Here's the phosphate group, which is yellow. And then there's your base, which is green. That's one way of looking at it. Here's a different way. It's the same molecule, just drawn a different way. Sugar in blue, phosphate still in yellow, and the base still green. And that's it. That's how it works. Any questions about this picture? It's just an example. I'm not going to show you this picture and say, you know, which part's the phosphate group, which part's the sugar, which part's the base. I'm not going to show you this and say, is this an A, a G, a C, or T? Nothing like that. This is just for your own knowledge. Like I said, each nucleotide has its own base, and the base defines the nucleotide, right? So remember, that's sort of like amino acids. Amino acids have their own side chain, and that's what defines the amino acid. And there's 20 different amino acids. But for DNA, there's only 4 different types of DNA. It's an A, a G, a T, or a C. Adenine, guanine, thymine, or cytosine. You do not need to know the full names. Probably not in the next exam, but definitely not in this exam. For this exam, just know the letters A, G, T, and C. And you might not even ask you that. But are there any questions about that? So again, just like there's 20 different types of amino acids, there's 4 different types of DNA bases. And here they are. There's a picture of them: A, G, T, and C. You can see that here that these are these have 2 rings on them. These have 1 ring. Not even going to talk about that in this chapter, but just keep it in the back of your mind for when we get there. Structure function, right? You can see that these have similar structures, but slightly different, right? These are slightly different than these. Therefore, their functions are slightly different. But nothing on this slide is important for this first exam. So what kind of reaction brings these 2 together? Just like everything else in this chapter, anything that brings together monomers, anything that brings together anything is called a dehydration reaction. So that's basically should be a review. And what happens is once you bring together these single nucleotides and you bond them to each other, right? You make those covalent bonds. Obviously, at that point, they are no longer monomers because they're together. So at that point, you get the polymer. So you need to know this for the exam. You already know because I said, what is the monomer of nucleic acids? The monomer is nucleotides. I'll put a big M here to remind you. I'll try. Monomer, nucleotides. So what's the polymer? That's the polynucleotides. And I like that one because that one's easy. That just makes sense. It's sort of like the saccharides. Monosaccharide is the monomer. Polysaccharide is the polymer. Same with nucleic acids. Well, sort of. Nucleotide is the monomer, and polynucleotide is the polymer. And I'm not going to get tricky. This is a multiple choice exam. So like one of your choices, I'm not going to say what is the monomer of a nucleic acid, and one of your choices is not going to be mononucleotide. Actually, I take that back. For most of you, that won't be the case, but inevitably somebody will miss the exam. And when someone misses the exam, as I said when we talked about the syllabus, they usually get a choice of having a 10% penalty per day that they missed it, or they take a slightly harder version of the exam. So that is one of the ways I might make it harder. But anyway, so again, these separate nucleotides come together in a dehydration reaction to make a polynucleotide. And when that happens, it's the sugar and the phosphate that are coming together. So remember, even though you didn't need to know this for amino acids, but hopefully you might remember, it's the carboxyl group and the amino group that come together to form those bonds. So with the nucleic acids, it's the sugar and the phosphate that come together to form the bonds, right? So it's not the bases coming together. It's the sugar and the phosphate. And that's where we get the name sugar phosphate backbone. And you can see that. I'll show you over here. There we go. So the strand right there, that's a sugar phosphate backbone, because we got the sugar in blue and the phosphate in yellow. Sugar in blue, phosphate in yellow. And that pattern just goes on. And you'll notice I skipped a slide. Let me go back to it. That's because for this exam, you do not need to know this. This will be discussed later. But the bases, well, let me go forward again. Here, what we have here are 2 strands of DNA. So when you see a DNA molecule, like you've seen this picture your whole life, I'm sure, what you're actually looking at is 2 different strands. So what keeps together one strand? The thing that's keeping together a single strand of DNA is this sugar phosphate backbone, right? Those are covalent bonds in the sugar phosphate backbone. So you have this one strand over here, sugar phosphate backbones, and then you have all these bases coming off of it, right? If you were to straighten it out. And then on this side, you have another strand, and that strand is that sugar phosphate backbone. That thing's kept together with covalent bonds, right? And then you have these, again, these things hanging off, these bases hanging off like rungs on a ladder, not doing anything. But then what happens is these rungs are hydrogen bonded to each other. So when you look at a molecule of DNA, like this picture right here, what you're looking at is 2 different strands of DNA. Each of them are covalently bonded. That's what's holding them together. But then strand A is held together with strand B by these hydrogen bonds between the bases. And I wouldn't have spent any time saying that because I'm not going to ask you anything about it on this first exam. But that will be very important later when we discuss DNA in Chapter 10. So are there any questions so far? Your book does ask some questions about this. A always goes with T. C always goes with G, right? But again, for this exam, you don't need to know it. You do need to know it for the study guide, but for the exam, not so much. Your book also points out that there's similarities and differences between DNA and RNA. And this will be important later in the semester, but for now, and for the study guide, but for this exam, I don't think I'm going to ask you any of this. Both DNA and RNA are obviously polymers of nucleotides. That part's the same. What's different, though, is the nucleotides. They're not the same type of nucleotides for the most part. Their nucleotides still consist of a sugar and a phosphate and a base. That part's the same. They're still made up of the three same basic parts. The only difference is some of the bases are different and all of the sugars are different. And I'll show you what I mean in the next slide or in some upcoming slides. Here we go. So again, the sugar is different for RNA. So for RNA, you have ribose as opposed to deoxyribose, right? Obviously, that's R for RNA, D for DNA, and it's also right there. That's where you get the name ribonucleic acid versus deoxyribonucleic acid. So there you go, different sugars. Not important for this chapter, will be important later. Instead of the base thymine, which DNA has, E, that's so horrible. That's supposed to be a D. Anyway, instead of having a D, or excuse me, instead of having a T like DNA has, RNA has a U, a uracil. And then finally, one of the biggest differences is instead of being double-stranded like that picture of DNA I showed you, right? That one DNA molecule was two strands of DNA hydrogen bonded to itself and twisted. And RNA is single-stranded. So any questions about this slide that will not be on the exam, on this first exam? All right, you guys are nice and easy. Here's another picture. This time it's RNA instead of DNA. Phosphate group's the same. Here we have a U instead of a T. Here we have a different type of sugar. It's a ribose instead of a deoxyribose. So this one has an oxygen, whereas the DNA doesn't. But that should tell you, again, structure function, right? The structure is nearly identical, but it is slightly different. Same thing with DNA, right? The structure is, the function is nearly identical to the function of RNA, but it is slightly different. All right, so every chapter has this portion, the process of science. I highly recommend reading it. It'll give you some good information that'll help you understand the chapter, that'll get your brain thinking scientifically, which is really the biggest important thing about this class. It's less about biology and more about thinking, how to think properly, how to think scientifically. So read it if you want. Basically, it goes back to this whole common thread of lactose intolerance, which was the common thread of the chapter. It's kind of interesting. I think it's really interesting. So what happens is humans are supposed to be lactose intolerant. Humans are only supposed to have milk when they're babies. But what happened was, and I'm only giving you the overview here, what happened was at some point in the human history, there was a mutation that allowed some humans to be able to continue to eat and drink dairy past a young age. So if you are not lactose intolerant, you are the person that has you're the one with a mutation. That's like me. I'm not lactose intolerant, so I'm the one that has the mutation. Just leave it at that. Yeah, and then later on, like I said, every chapter also has this evolution connection, and every chapter I'm going to skip it, but I like it because it goes along right along with this thing that we talked about, the process of science, where I talked about this mutation. This talks about evolution. It's really neat. I'm just going to talk about it really quickly just because I find it slightly interesting. 80% of this is from your book. It says 80% of African Americans and Native Americans. If I were writing that, I would say 80% of people from direct African descent, which are African Americans, but also you think about an African person living in England or Jamaica, right? So that's why I don't like the word African Americans in this context. But anyway, so 80% of these people, 90% of Asian Americans are lactose intolerant. So only 10% of people from like Northern Europeans, right, Caucasians, white people, if you will, only 10% of them are lactose intolerant, which is interesting because what probably happened was in evolution, these people that are still lactose intolerant, because they live in hotter areas, depending on where they were, they probably weren't storing dairy, right? So they didn't have like, oh, here's a cow, I'm going to milk it and keep this milk around for a few days, to feed my family. Can't happen in sub-Saharan Africa where it's hot and humid. But if you're living where it's nice and snowy and cold, you can milk a cow and put it out in the snow, and and you could have milk for a while, just like we do now with refrigeration. So because of that, that mutation probably popped up in somebody in Northern Europe, and that actually was an advantage, and therefore it passed along. And now we have more people who can drink milk of Northern European descent. Anyway, you can read all that if you want. None of it's going to be on the exam. That is the end of chapter 3. So, shoot, we only have 9 minutes. We don't really have time. How about this? Instead of doing like a really normal exam review, which is where I take your very specific questions and I pull them up on the screen and we look at the questions and I answer them. Does anybody have any basic questions about the exam? Like nothing specific about the content, because we'll discuss that tomorrow when we have a real exam review. But does anybody have any?
Will the exam be like multiple choice?
Yes, great question. So here's how the exam is going to work. You're going to log on. If you're not in person, if you're not taking an in-person paper version, you're going to log on to the computer like you did this morning. You're going to turn your camera on, but you're going to stay on mute. That way I can see you, right? I can see Thornabar over there. So if he were to like pull up, if he was taking a test and I see him doing this, I'm like, I can see him. What's he doing? He's cheating, right? So that's why I need to see you. But anyway, so log on to your computer, turn on your camera so I can see you, and then keep an eye on your email. So I'm going to have the exam. It's going to post. It's going to be scheduled to post a little bit before 8. So as soon as you see it post, you open it, and it's going to look just like your study guides. And this is why I keep telling people, do your study guides, because when you pull it up, it's going to look, just like this. So you should be familiar. If you do the study guides, at least then you'll be at least familiar with the layout, right? Because it's going to look just like this. You can understand how to navigate this and how to answer the questions. So yeah, that's how it's going to be online. And then it needs to end at 8:50. And I'll try to remind you, because we'll all be online. I'll try to say, hey, it's 8:50. You need to log out. But ultimately, that's up to you. So you have to log off at 8:50. Any time you log off past that, or don't submit past that, you're going to lose points. Does that make sense? So you have to log on. You have to start it. You can start it as early as you want. As soon as you see it pop up, I don't care if somehow there's a glitch and it opens up at 7:30. You can, as long as you're on the computer and I can see you, you can start it as early as you want. But you have to finish by 8:50. Again, you can do that in here if you want. You can come into the classroom with your computer and still do it online if you want. And then the third option is you tell me, like one of you already has sent me an email. Let me know today because I need to print it today. I will print a copy of it, and you can come in here and I will give you a physical paper copy. Oh yeah, then the fourth option, if you know you're just not good at testing, you're like, well, 50 minutes isn't enough time, I will give you more time. You just need to contact me today, and we'll find a time to meet online like we are right now. But instead of the whole class, it'll be just one-on-one, and I can see you and you can see me. And then this time you'll have your microphone on so I can hear you, and you'll share your screen with me so I can make sure you're not cheating. And when we do that, I'll get to you in a second, Braden. Thank you. So yeah, if you need more time, contact me and we'll find a time to meet. And as long as I can see you and watch you like one-on-one, almost like me sitting over you watching and taking an exam, you can take as much time as you want. So Braden had a question. What was it?
Okay, so like, how many questions, like, is it gonna be like a really like big test that we would be able to finish in like 50 minutes?
It should be about 50 questions. Another reason to look at the study guides, because the questions will come from the study guides and they will be slightly reworded. So, and again, the reason that's important, one, if you do the study guides, you'll be familiar with what the questions are, even though there'll be different versions of them. And two, like, I don't know how good you are at reading, right? So if you do the study guide and just try to imagine in your head doing 50 of those in 50 minutes, then you'll know, it's like, yeah, that's not enough time. I should contact him and work something out. Or you might do it and be like, oh, that's plenty of time. So great question. Yes, there will be about 50 questions, give or take. It should be about a third from the first chapter, a third from the second chapter, a third from the third chapter. And yes, to remind you, there will be a few questions from the syllabus, because I keep saying read the syllabus, right? And obviously it's a really thick syllabus. I don't need you to read all the stuff that you would get from every single class. Read the parts that are important to our class. Yeah, that's it. So any other questions about the exam?
I have a question.
Yeah.
So it's more about if we think we may need more time. So we contact you today, and you'll schedule like a time on Friday, the test day.
We'll figure something out. It can be a little bit earlier. Basically, I'll do it probably, I don't know, maybe until Sunday. We'll have a whole week starting like today to Sunday where you can have, we'll pick a time. Because normally I would say that's unfair. Like if everyone's taking the test on Friday, then other people are taking it, on Saturday. You could be like, oh, they had an extra day to study. But the thing is, anybody who takes it with me, it's almost impossible to cheat because it's one-on-one. I'm watching you. I'm listening to you. I'm seeing your screen, right? So that's the only reason I even have timed exams. Well, first of all, we're under a time crunch because we have to be done at 8:50. But other than that, with online exams, that really prevents people from cheating. Even if I said, just go ahead and take it and don't even log on, with 50 questions at 50 minutes, you don't have time to look up every question.
Right.
So yeah, I'm fine with doing it on a Saturday or Sunday. Obviously, I like to enjoy my weekend, so I prefer not to do it on a Saturday or Sunday, but I'm here for you guys. So if it needs to be done on a Saturday or Sunday, we'll work something out.
Okay. But if we do the study guides and like, it should be doable in 50 minutes. Right?
I think so. Yeah, and it all depends on what kind of reader you are and what kind of test taker you are. Like I can't answer that question for you. Some people are quick readers. Some people are very slow. Some people have test anxiety, which normally they'd be really quick, but that just bogs them down. So it's a very individual thing, which is why I'm giving this opportunity for anybody who wants to do it separately and get more time. Any other questions? All right. So before I stop recording, I'll say this. Obviously, we didn't have time to do the exam review. So sometime tomorrow, I will do an exam review. I'm going to post something on Google Classroom. So you guys keep an eye out for that. And then you just respond. So if you prefer to come and actually be involved in the exam review, then make sure you respond and say, hey, I can be there at 10, but I definitely can't be there anytime after that. Or I definitely can't be there at 1 p.m., but any other time I can be there. So let me know once you see that. Otherwise, I will see you for lab today in this room at noon. Make sure you're wearing closed-toed shoes and pants. I guess that's it. Any other questions? All right. Oh, yeah, make sure, I know I got a lot of you for attendance, but if I didn't get you for attendance, make sure you email me and say that you watched the video. I'm going to say there's a keyword, so I know that you watched it and just not saying that you watched it and didn't give me the keyword. Today's keyword is going to be clock. So send me some sort of email and say, hey, I watched the lecture today and the keyword is clock, and then I'll give you your attendance points. All right. Everybody have a great day. I will see you at lab or during office hours here in 10 minutes.